Huntington's is a genetic disease caused by a mutation to the P arm of chromosome 4 at band 16.3. It is caused by a trinucleotide repetition where there is an excess of repeats in the CAG sequence typically found in a normal HTT gene. This produces a stretch of polyglutamine which forms a misfolded protein. Huntington's in humans causes a progressive breakdown in neurons which affects an individual's ability to move and think properly. Symptoms of Huntington's disease include muscle problems, slower abnormal eye movements, difficulty with speech, and other neurological problems. Currently, drugs such as tetrabenazine and deutetrabenazine are the best treatments. Both of these drugs suppress chorea, which is another main symptom of Huntington's.
These drugs, however, aren't going to cure the patients. We decided to use the PCR technology to isolate and study the gene for genetic cure. First, the restriction enzymes NHE1HF and APAL1 isolated our gene. Next, the DNA was denatured. Once separated, our primers marked the replication points on each strand. Then extension took place when Taq polymerase replicated the gene. This process was repeated many times, resulting in many copies of HTT gene. Finally, we used electrophoresis to isolate the copies of our HTT gene, which is 1194 base pairs shown here. After obtaining enough copies of HTT gene to work with, we came up with ways to genetically cure the disease. First, CRISPR technology could be used to replace the excess CAG repetition with the normal amount. First, we came up with two Cas9 proteins with two guide RNA strands that will bind to before and after the CAG repetition. After recognizing the PAM site, each Cas9 proteins will cut the gene before and after the mutation. Lastly, the donor DNA will replace the cut mutation to genetically cure the gene.
Since Huntington's is found in an autosomal chromosome, somatic in vivo would be our preferred gene therapy type. For our vector, the virus adenoviridae would work well given its broad host range and flexibility with varying cell types. We can remove the viral material and replace it with our new CRISPR cut sequence or with microRNAs. MicroRNAs prevent or reduce the production of particular proteins. This way we can have the microRNAs bind to and inhibit the production of the mutant Huntington's protein. Either way, we can directly inject the vector into the glial cells, which will reduce the production of HTT proteins. Over time, this could cure the patient.
Lastly, we wanted to test if our CRISPR editing worked and the HTT gene of the patient was fixed properly. To achieve this, we obtained 3 samples of HTT gene and 2 types of restriction enzymes. We compared the gene from a healthy patient, our mutated patient, and our fixed sequence. Then we used 2 restriction enzymes, HPYAV and ACI1, that would cut the HTT gene before and after the CAG repetition. We ran our enzyme cut DNA through gel electrophoresis. As a result, we could see that our CRISPR treatment worked. The well with the normal person's HTT gene and the well with CRISPR fixed HTT gene look identical, meaning they have the same genetic material. Additionally, we could see the polymorphism of the HTT gene is present because the patient's HTT gene before CRISPR had one gel band that was different from the gel band of the normal person. Because the CAG repetition is longer for Huntington's patient, one gel band was larger. The fact that this longer band was lacking in our CRISPR cut gene proves that we have shortened the harmful trinucleotide repetition to its normal amount.